we're good to go. Hi, everyone. So thanks for coming to the hackathon. So now we're going to share the results of people's work over the course of the last 2 days. And we're not expecting to have finished anything. This is very much an exploratory exercise to just generate ideas, get people talking to each other. So if the team from CASM would like to come up. And if at any point anyone's got any questions, I'll run over with the microphone and then I can shout them out.
Good afternoon, everyone. So here we are going to present our achievement in this hackathon. The project we proposed was using machine learning AI method to do laying QC. Essentially, we have big tabular data and doing binary classification. We take in this tabular data, feed into the model, and the output are expected to be either A or B, or 0 or 1. At the beginning, the first challenge we met was trying to do some data cleaning, because initially the data we proposed turned out to has some flaw in it. So we divided the team into 2 bits. Part of the team doing the data cleaning and part of the team doing the model building, so we can run in parallel without blocking each other. When the data cleaning finished, then we do the model building. Now I'm going to hand over to one of our team members, talk about data cleaning. I mean, these people do the actual work, please.
I didn't actually do it. You did it for me.
Go ahead.
All right, so the initial data set lacked the context needed to actually do the lane QC. So what we did was we identified all the current methods of evaluating the lane QC and how those values are calculated. Then we dug a bit deeper into our database to generate a data set that we could actually use to train the model. From there, we refined the data set a couple of times, basically running through quite a few data sets till we got one that we were happy just to start feeding to the model.
So we mentioned about we are dividing the teams, and part of the team doing the model building, and we're actually building 2 models. I'm going to briefly introduce the model 1 we have built. Initially, we do not really know what the information can be contained in this data set. So we proposed to do like 2 model building. One, the first, like as you can see from here, the first model is slightly narrower, only contains 3 layers, but with like a slightly fatter, that's not quite true. This maybe should have more neurons here. So it's a kind of a more wider, but shallower shape of the model. The other thing we were thinking of was if we can further extend the number of layers, would the model can capture more of the hidden features within the data set. So we also built another data model, which contains a more number of layers, but supposed to be each layer contain less number of neurons. But anyway, so we run the same data sets through both of the models, and then we have some kind of a rough idea of which model can give better results. Then we use that type of model to do the training. So the other thing I want to mention is the normalization effect. We talked about data. We did the data cleaning. But even after cleaning the data, we feed into the data model. We still have some problem in making the model can read the data properly. For example, in some of the cells, we contain like a really, really big numbers, like many thousands. But while in other cells, we only contain numbers something between 100 and 200. So before the normalization, we feed into the model. As you can see from here, the white model only delivers something like 50%, which wasn't very ideal, because if you think about the binary output, it will be either yes or no, or A or B. It's by default it's 50%, so it didn't help much. And the deeper model gave slightly, roughly the same, probably even worse results, and didn't help. Sorry, this thing is not quite accurate. Apologize for that. Ignore it. Then we think, okay, maybe the model giving too much weight for the big numbers, but and didn't give enough weight for the smaller numbers. So we did the normalization across different columns. So we compress everything from no matter what the original numbers are, we compress them into the range between 0 and 1, then feed into the model and do the learning. As you can see from the right-hand side, the results are much better. Here, the white model gives something 88% accuracy, and that was also very consistent. And the deeper model gave even better results, as you can see from here. So this is something we want to mention. Sometimes we don't see some feature, we don't see the model perform well. Might not be the problem of the model, it's how you talk to the model. The next one, I'm going to hand over to Yichen, talking about the second model we built.
So basically, we split into several subgroups and then design different models to tackle this problem. So the second model we try is not a neural network, but instead like a traditional machine learning method called Adam Boost. And the concept is built on decision trees and random forest. So for random forest, like when you get a new samples, you get a group of decision tree to kind of looking at its features to decide whether it's class 1 or class 2, basically. And then after all the trees have their predictions, there will be an averaging of their report, and then you will get the final prediction out of this random forest. And then Adam Boost is built on this concept with one improvement, which is for all the decision trees in the forest, they are not independent trees, but instead they are built on the foundation of each other, which means for tree 1, the label on the samples that it was mislabeled will be assigning higher weights in the second tree in that forest. Therefore, like each tree is kind of gradually improving the results of all its procedures, and then kind of aggregating the power of those tree predictors to achieve better results. And I think that's the previous group. They need to improve the model performance. And then for these methods, I think there are mainly two approaches to tweak. One is the hyperparameters of the model. Compared to neural network, it doesn't really have lots of parameters. So they are basically the number of trees you used, and also the weight of each class, because this is an in-bandness data set. And then another way is to like kind of improving data set itself, which is to bandness the data set instead of using the original data set. And then, so I'm just showing the best results, which is worked on the bandness data set. So the data set together consists of like about 30K whole genome sequencing length QC data, and half of them are passed, and then half of them are failed. And then I use 70% of data for training and the rest for the validation. And I think we achieve similar accuracy and of the results. And then when we break it down, it's kind of the results is also kind of very balanced between the false positive and false negative. And therefore, it's kind of, if we are translating it to the plain language, it would save over 90% of manual effort to look at those in the labels. If you just want to highlight the majority of the fail cases, which is or also over 90%.
So for the samples that were misclassified, do you have a feeling if they were the same samples that were misclassified by the previous model? Or is there just some tricky samples, or is it just random?
I'm not sure about that. So we didn't really compare, but we can. I think it's worth looking. And then also one advantage of this kind of model is that it's not a black box model, it's a white box one. So you can actually just look into the model and to see which feature is actually using for prediction. So for these cases, I think the top features it uses is the percentage map, and insert size, standard deviation, CACs, duplicates rates. I'm not sure if it's the same with what we are using for the manual creation, but that's how this model automatically learns for us.
We have one more slide. Sorry about the order of the slides. We have a thing about improving model performance.
Yeah, this is I think what we did for like balancing. So yeah, I think one thing worked was like balancing number of fail and the right cases. So earlier we were using the one which passed, but I think the accuracy was not that great. So that's what we tweaked, and we worked with neurons, trying deeper, wider models, and just trying different activation functions. So I think that's what, yeah, we're just seeing what works and where we get more accuracy. So yeah. Yeah, that's all.
That's everything we are hoping to present, and it has been a great experience. And here's the team. We have another team members, which is Martin, and he's preoccupied by something else, so he couldn't be here today. We have also team members that put a lot of input into this, but didn't get a chance to speak much. So yeah, feel free to add anything. Or any questions?
Did you train the model on the uncleaned data?
No, because it's the one reason of that, because it's more of like garbage in, garbage out thing. If the data contains something irrelevant with the output that only polluted. For example, one example will be in the historical QC data, normally we will have a record of who finalized this decision. That would be like a single username. If we include that kind of information into the training, then you're probably getting, okay, this person historically likely to approve something, that person might not. So it's kind of polluting the data set. Did I? Okay.
I'm interested by your model architecture choices. Was there any particular reason that you wanted to pick the certain number of neurons in each layer, or was it just?
It's pretty arbitrary. I think that the input layer and output layer somewhat is going to be fixed. The other day, I borrowed some of your concept using the big prime numbers, and we tried. It seemed to be working fine. Initially, we used smaller numbers. Then we found the result wasn't very ideal, because thinking about only 3 layers, if you have a smaller number of neurons in the middle layer, then the out connection you can make somewhat can be restricted. So we increased bigger and make it a much wider model, and that could potentially capture something. Also, similar thing with the deeper model, because we look at the say number of columns, I don't know, 20 columns or something. You think about maybe these 20 columns can give us all the information, but sometimes if you do some correlation or somewhat kind of conversion, something can be explained by the model but not ourselves. So I'm thinking deeper model could potentially surfacing those features and help us deliver better accuracy.
And if you had a lot more time to put towards this, what would you?
One thing we have partially done is trying to normalize everything in the middle, between the range 0 and 1. The other thing I would like to try is, I think I will discuss it with the team also, is something like, because if you look at the numbers, they're relatively in small range. I was thinking if we take a square of each number, can potentially making the data point like in a much bigger space and more sparse. I don't know whether that could help or anything. Another thing we would like to try, at the moment we're using ReLU function and sigmoid. That's very generic. We haven't got time to try whether other activation functions can give us better results in this particular case. that's something we might want to try. I probably will be.
Out of interest, has anybody used any less used activation functions? Okay, so
I used to use some different activation functions and use different initialization for the network.
Mm-hmm.
But I found it doesn't influence too much, because sometimes if the model is simple enough and the data set is not very complex, I don't think this will influence the results very much. Yeah.
any more questions? Well, thank you very much.
Thank you very much. Cheers.
Thanks very much, guys. So next, shall we go for Gen team? Or I don't know what you want to be called.
in your laptop. Yeah, that would be great. Yeah, if you can introduce them.
Okay, so we don't have slides, although I might draw some things. Oh, I have to go near the right mic. I won't draw some things then, I suppose. So we've been working on a set of these MAVE data types I introduced at the beginning. For anyone who missed that bit, it's what we were working on is basically a matrix of fitness scores for all 20 different substitutions that you might make at each possible position along a protein sequence. And we've got a data set which has a whole load of these experimental data sets. It's sort of 2,000 studies across about 1,000 different genes. So it's sort of moderately big in terms of, well, it's big in terms of the number of experiments done. It's not super big in terms of machine learning data sets. And one of the other features of it is because it's coming from experimental data directly, there's quite a lot of missing values, and it varies between the studies. Some of them have quite a small number of real measurements, and there's actually a lot of missing stuff. They're maybe not that useful. Some of them, there's just a few kind of missing bits scattered around. So one of the first, I mean, we started off just discussing different things that we could do with the data type. One of the first things you need to do, though, for a lot of stuff is impute or fill in some way what these missing data points are. So that's what we kind of set off doing. We kind of split up into different approaches because there's lots of different ways to do that. Some people who I think are not able to be here to present today did some sort of baseline benchmark stuff using kind of mean and medians that you generally see in that column or row, and compared that to the what you actually get. I didn't see their results, but from other data or papers, I know that works moderately well. Then Eric has been doing something slightly more complicated and a bit more sort of data-driven. And then today, she will present in a second, and me and Ronnie have also looked at a sort of more machine learning-driven approach, trying to use embeddings from protein language models to impute the missing data, which I suspect will work. We've got it running in some sense, but it turns out it's going to take ages to do the ESM scores on my laptop, so that might be a job for the compute farm, and I'll get back to you in a week or so or something. We also discussed, because it's a data matrix, which is basically an image in terms of machine learning, using these diffusion modeling approaches would actually fit very well here. So you kind of add a load of noise to the data and try to iteratively denoise it. And for generating pictures of cats and pictures, not pictures, but like regenerating data tables, this is known to work well. So I suspect that also will work well, and we discussed quite a lot about it, but it's much more technical than you can do in this kind of 2-day period, given that there's not an off-the-shelf package. So, which is why we went for this language model-based approach, but which is also too heavy. So what we do have, though, is Eric's approach, which he can kind of talk you through now.
So I got a bit lucky. I found there's actually a scikit-learn package that runs the input as a model that you can select multiple estimators. That means it's a API you can plug in with different types of models, estimators, and play around to see which one actually gives the correct answer or closest answer to what we're expecting. So just given a quick demo of what the initial iterative imputer is doing. So I got this data set. I've done some data cleaning to start with because some of the runs have too much NAM values inside those experiments. It's probably not a great idea to impute those. So what I found was there's a distribution of 20% of these values are missing. That is most of the experiment, but some of the experiment has quite a high proportion, so I just collect everything below to 20%. The second part is think about 20 amino acids. If you mutate one amino acid to itself, it's mostly having no effect. So in this case, the experiment wasn't done, so most of them are missing, but it makes sense just to fill them with 0. So after those cleaning, we got this initial run just using the default estimator provided by scikit-learn with all the default hyperparameters. And you can see these little white squares that are missing values got filled afterwards. So that's a pretty good initial success because the entire thing takes about less than a second for one single experiment. So because it's statistically modeling based, I would expect it's not as time consuming as a lot of machine learning techniques. I haven't looked into the interpretation of what the parameters of that, but that's something else we can try after we select a good estimator. The second step I've done, don't know if I can demo it live, probably not a great idea. I'm eager to try it. The second thing I tried was to actually run this iterative imputation for 4 selected imputers. These are, I have one of the functions running it. These are Bayes and Ridge, which is the default, random forest regressor with some hyperparameters already set up, a NeoStream pipeline with Ridge. So it's essentially 2 pipelines connecting together. And finally, the KNN nearest neighbor regressor. So if I've got that running, you can see this entire thing is doing the training and evaluation. And you can see the progress of running all 4 different estimators. So for evaluation, I take the test data set and training data set with a 20% to 80% split. And in those test data set, because we still don't know what's the actual NAM value is. So what I did was I artificially introduced more name values to replace the values that are in the original data set, and then to see if my imputed values of those actual name value is going to fit my prediction or not. So that's kind of running on one of the experiments I selected, deliberate selecting one with 300 measurements. That's like larger samples in this entire data set. And you can see there are some differences between how good they perform. The first column is I measure the mean square error, which is the kind of most naive way of evaluating a model. And the second and the third are a Pearson correlation and Spearman correlation. The higher the correlation, you expect the model to behave more consistently, predicting the actual outcome fairly well. So there are some methods that actually stand out or are better than the others. The original default, Bayes and Ridge, is actually not doing too bad. So that's a good thing to know. Perhaps we're just going to stick with the default to evaluate a larger number of experiments. There are a few next steps. First, I found out this approach is not able to handle when all the mutations in a particular amino acid are non-numbers, because it's statistically impossible to impute. You have to hallucinate to get the number. So linking to that, we could try to combine data from different experiments. Different experiments will have a different protein target, so that's something we need to be careful. However, we believe in statistical power, so having more experiments would potentially give a reasonable results. It's all can be evaluated. There's also very promising approach of using boosting. I just realized it was mentioned in the previous group presentation. Boosting can be a good strategy of combining multiple estimators, trying to take the good ones, absorbing the better statistical power out of multiple ones. It's also suggested by one of my teammates as well. So it's definitely a good strategy to try. And finally, it's good to mention that the wild type amino acid before the mutation is probably something important, because if you imagine a hydrophobic amino acid mutating to a hydrophobic one, it's going to have a less effect on the overall fitnesses compared to a hydrophilic one turning into a hydrophobic one. So the initial feature is definitely something we can concern. The question is how to actually incorporate into a model. We're thinking about like the approach that Ronnie and Ali are working on, about trying to embed these informations about amino acid and introduce that information into the model. I was also thinking about getting more columns. Each of the column, instead of just mutating into one amino acid, it's going to be from what amino acid mutate to another amino acid. It will make a much wider table. The question is how to actually combine that, how to define each of the observations, because a sparse table probably is not very good to work with. So there is some strategy to try following that approach. So yeah, it's a lot of things to put in the next steps. Perhaps more hackathons to come in. But any questions for us? Please.
Thank you very much. I was thinking, Eric, this is a really good demonstration of how, although it's tempting to use the latest and flashiest AI model, just keeping it simple and keeping it statistical is often, you're getting pretty good results just using fully explainable statistical models. I wondered if you had any sort of intuition as to when is the right time to sort of step it up to a larger AI-based model, or when's the right time to pull it back to more bare bones, if you like?
I think a good inspiration is that if an easy model, a simple model works, stick with the simple one, because the simple one is less likely to, I don't know who said that, but it's less likely to make mistake than a complicated one. But secondly, it's also from observation of this entire data set. It's not a huge data set, according to what our observation and initial exploration of the data structure. If you look at each individual experiment, it's only about tens to hundreds of records or samples pairs that actually get into the data set. In that case, I would say a machine learning model will potentially struggle because of the lack of the training data. If you got more data, machine learning model would perhaps behaving well because it has more layers. It could incorporate more information into the nodes or the weights of the machine learning structure. But a statistical model tends to, I would believe it more for a smaller number set.
And actually, perhaps coming off that, Ali, I wonder if you have any thoughts on how, as the size of AI models increase, and we're expecting larger and larger data sets, there is a sort of natural limit to the size of the protein space that exists in the natural world and the genomic space that exists in the natural world. Do you think that the data will be able to keep up with the requirements for these models, or do you think we're going to have to find new ways of generating synthetic data and other ways around those limits?
Yeah, so I guess it's a good observation that there's a lot of possible proteins if you sort of think in theory about the number of like strings of amino acids, but the ones that you actually observe in nature are quite a tiny proportion of that. And we've probably already seen the majority of them in our sequencing databases, at least the majority of sort of classes of protein, until we like delve into sort of weird archaea in a hot spring or something, and then we might find a new group. But we've mapped most of the kind of standard proteins I would expect, and we wouldn't expect to kind of get more data really. So in some sense, this is probably what we have to work with for a while for building the models. We can keep making UniProt bigger, but most of them will be, very homologous to something that's already there. So there is going to be a limit of, as you just make the model bigger and bigger, it's just going to be learning what's in UniProt, and that's not actually very useful. It's sort of a glorified lookup table. So there's various things you can do. One of them, which I guess is the generative genomics to plug our new program, is create new variation and then measure it yourself, and you can create that variation kind of out of distribution from the wild type space and then measure what are the properties of it, and that's something that some of the labs are interested in doing. But those kind of experiments are always going to be lower throughput, or at least more focused throughput, than just kind of trawling the sea and sequencing all the microorganisms you find. So it's quite a different type of data. The other one is doing these kind of MAVE studies on known proteins or protein families, and there you get variation around the existing sequence, and you can do either like all the singles, or you can try and push out into combinations of mutations, which some people are doing. But again, you're limited by the throughput of your experiments, and that's always, while you can get them pretty big at this point, there is just a limit to how many cells you can grow in a vial, or how many bits of DNA you can have in a tube meaningfully, compared to the number of possible combinations. So we need to, I guess, design models well so that they're actually understanding the biophysics or functional properties of the protein sequences rather than, I guess, just learning what is out there if we want to get beyond this paradigm of just getting kind of very good performance on the existing natural variation, which is, I'd say, where we are now, but which is useful but doesn't necessarily explain all the things we would want to know about, for example, designing new protein spaces. Although we do start to get reasonable performance there with some models that exist today, I suppose. Yeah, I guess that's my
Thank you.
monologue on that topic.
Any other questions? Well, thank you very much, both. Thank you again. And then finally we have Sean and his group. You ready to? Nice.
Okay. So do you have any idea to just display that? Display mode. Oh, okay. Hi everyone. Actually, we are super excited to let others know we made some progress in this project. It is really amazing. So actually, the auto curation project is from one of my projects I'm working with. Yeah. So I'm Sean. You can call me Sean. My Chinese name is Shaoheng Guan, and my teammates.
I am Sean. It's not very similar, but actually quite different.
Yeah. So thank you very much for this project. Yeah. So actually, I'd like to introduce the background once again. So for the reference genome we uploaded to the database. So before uploaded the reference genome to the database, we need to do a lot of work. Okay. Okay. So before just the uploaded of the genome to the database, we need to do a lot of work, like the wet lab work, and then run the software to join the reads to contigs, and then run the scaffolding software to join the contigs to scaffolds. And even we got the scaffold genome, we still need to do curation work because there's some small contigs can be misjoined or just left aside. So that's why we are doing the AI curation project. Yeah, that is my project, but for Hackathon, I just collect some of data from the project and do the Hackathon here. So for the high C curation, it can simply be explained as this. So the dense point here means that this fragment has very dense interaction with them. That means this fragment should be physically close to this one. So we need to move this fragment to here. Yeah, that is the simple description of this project. So we can see after the curation, in the other side, we got a curated Hacking map. That one is much more tidy. Yeah. So this project is to use AI to do this automatically instead of do that manually one by one. Yeah. The AI can read all of the Hacking map and do this just at 1 second. Yeah. So the method we are using in this project is a graph network. We just represent every fragment into a node in this graph network and use a link. You can see the W15 is to link the fragment 1 and fragment 5. Yeah. For the edge, we have 5 attributes. We have 5 attributes to describe how to link these 2 fragments. Because for genome fragment, we can link the head to head, head to end, and end to head, end to end. So there are 5 kinds of link method. Yeah. So first we just build all of the fragments in a sample as 5, not 5. How many fragments in the sample? We got how many node in the graph. Yeah. And then we use the graph network. Specifically, we use the graph convolution operation. Yeah. After the graph convolution, we got the likelihood matrix. Yeah. So, for example, if we have 500 fragments in the sample, then we got this likelihood has with 500 lines. Each vector is going to represent 1 fragment. And then we're going to use a fully connected deep network to predict the likelihood to link them. Yeah. So that is method we are going to solve this problem. Yeah, and then this is the prediction given by the graph network. We just predict the linkage likelihood. For example, 0 to 15 and 0 to 15. That means there are 15 fragments in this sample. So we need to calculate the likelihood between each 2 of them. So we got this matrix. And then we got this matrix that, for example, for this point, it means we need to link like around 13 to 12. Yeah? These 2 fragments has linkage to each other. That means we need to link them together. And then after we got this prediction, we just use a deep first search. So if you ask what is deep first search, I'm going to just introduce it very simply. Yeah. For this one, I'm going to start from the number 13 or 14. And I just search here. Then I got the 12. Then I will search 12. This is also 12. In this 9, this is all the fragments can be linked to 12. I just select the best one and link it to 12. This is just like in the binary tree. Yeah. For S has 2 selections for S. We just compare A and H, which one is the best for S. And then select A, and then we got to A. We just compare B and C, which one is best for B and C. And then we select C, then we select E. Yeah. So this is the deep first search we used to search for the.
Just wondering, does it matter then when you start to get different results? So just wondering, does it matter where you start in the search, or will you always end up with the same results no matter where you start?
Thank you. Very good question. Yeah, actually, I have already modified the deep first search. Just I can search from the head and also from the end. And then, yeah, actually, these were influenced because this is like shortest path problem. Yeah, we need to find the shortest path to link all of the node. So for this kind of problem, there's no accurate answer to that. The stars will influence that. Yeah. Yeah, but hopefully we are going to. This is not the main problem for this question, I think. But it is very important. Yeah, thank you. Yeah, this is a deep first search. So actually, for this hackathon, we are going to do some model improvements. For example, we need to find the red chromosome for the small fragments because only the small fragments cannot be joined. And then the second one, we are going to use the deep first search. Actually, during this program, we just look into the deep first search and yeah, for the deep first search, if we just look here, for S, we just search for the end of S, but actually in genome, for one fragment, we have an end and we have a head. We need to search both of them and to find which one should be linked first. Yeah. So here we also made some modification. Also made some modification to the deep search first, and then because of the overfitting problem, we just expand our data set. And also Shuang is mainly working on overfitting problem. Yeah, so you can, maybe you can. Okay.
So before the slide, we also have one slide for describe the data that we're using for the hackathon. So do you want to go through that first?
Okay. You want to introduce the work you've done first, and then when I go to that slide, you can introduce that specifically.
Okay. So basically here, just to repeat what Sean has been saying, that we mainly wanted to solve two different problems. The first one is to generally improve the model performance, and the second one is to alleviate the overfitting problem that we have seen in the original model, because the model is actually quite complex, and then we have a very small data set, and somehow it's still noisy, and that's why we are that's what we're doing for this hackathon. And now is actually the data that we're using for this hackathon that's based on the human T2T genome.
Yeah. Okay. So actually, the data preparation is not a part for this hackathon, because I've already prepared all of the data and uploaded data into Google Drive. So this is the data we are using for this hackathon. If we are using the data set from Tree of Life, that data set can be much more noisy than this one. So we just collected the human T2T genome and collected the Hi-C map as the ground truth. As you can see, here we got 6 chromosomes. Yeah, we got 6 blocks, which each block of them is just the 1 chromosome. And then what we do for the data preparation, we just cut them randomly. As you can see, we run the random cut. Yeah, for example, this chromosome is just cut it into 1, 2, 3, 4, 5. Yeah, cut it into 5 pieces. And random cut, and then shuffle them. Yeah. In the right side, this is the Hi-C map after the shuffle. After the shuffle, we just get a random Hi-C map, just like the Hi-C map before curation here. So yeah, here is the data set this is the real work. So this is the real work from our Tree of Life team, the curation team. They are going to move nearly all of the fragments to where it should be. It is a massive labor work. Yeah, it is very complex to do this kind of work. So for this kind of work, it is too complex. So we are going to generate some data sets from the human genome, just like this one. Yeah. After shuffle, we can see this is a little bit like that problem. And then we are going to work on this sort of problem based on the human genome. So the overfitting problem.
Yes, thank you for that. So we know that we wanted to predict basically the likelihood matrix based on the previous Hi-C data we see, and on the leftmost is actually the original model performance. Like I only plotted the MSE for both the training and also the validation data set, and in the x-axis you see the different the number of epochs. That's no, the number of iterations, I think. And then you see in the original data set, it doesn't really get much trained, and also the overfitting is like pretty severely. And so I tried 2 different strategies. With the first, actually the most straightforward ones, I used a bunch of batch normalization for all the layers in that network, and also I added like L2 normalization with a very small like lambda for the L2 normalization, because once we have a really big lambda, we just see the model just doesn't really learn at all. And then you see like it's somewhat better as it like, and also And actually, also surprisingly, it helps with the, I think it's mainly the batch norm layer that actually help with the overall performance also be a little bit better. So that's with the batch norm. And the second thing I tried is that I tried to implement this this variational graph auto encoders, but it's actually not really an encoder because I don't really auto encoder because I don't actually have a decoder there. I just basically have the KL divergence plus the MSE as the loss function there, and so that I still have the benefits of adding this regularization for the latent representation for the nodes, and I wouldn't say it's like working fully, but and also I honestly think the performance you see here could also be some of the details I had in the network because, for example, I still have the batch norm in the model, which could or could not, which should or should not be actually in the model, and I also have like some activation function matrix, a function that that maybe needs some more consideration. So, but for now, that's what I like been doing for the hackathon, and I would basically say like both these strategies probably helps with the overfitting, and to some extent helps with the general performance. Yeah.
I'm going to introduce. So we also did some experiments just on the overfitting problem. For example, if we just use a neural network to predict x to y, x is just x is like the coordinate value, and y is just the sign x. So if we just add some noise only to x instead of add the noise to y, we can see this one is just to get overfitted on the training data set because we didn't give enough data set for the right value. We just need the model to predict, oh, the y is the right, then give me the answer, train it. So we can see the model is just to get overfitted on the training data because it's just going to give the right answer instead of find the knowledge behind it. The knowledge behind it is just the sign x. Yep. So if we did another experiment to show the overfitting problem. This is very interesting. I'm going to show you. So, for example, this one, we can see. Yeah, we can see this one by one. So here, the x value, the real x value is just the Nimbase from 0 to 2 pi. Yep. And I just add noisy to the x and add more features to the noisy one, more features. I just add the random features to add, yeah, for number of features, I set this as 3. So I just add some useless features to the input side. And then I'm going to run this network. If I just add more, for example, here, the noisy one, yeah. Only the first term is useful. The second term, the talk dots random neck, yeah. This is a totally random. This is a totally noisy feature, which is useless. But if we just run this one, let's see what will happen. Yeah. Yeah, we can see the model get overfitted, even more severe. Can you see that? So this is a loss on the training set. This is a loss on the validation set. We can see the problem even get more worse because the network just trying to give the right answer, even to find the clue from the noises. Yep. So that's why our model is getting overfitted. Actually, one of the problem from the data set. Yep. Okay. So let's continue to work on some. Oops. So for the next one, yeah. So after our experiment and after our trying, yeah, we train them after the modification. We just train the model on 1,800 samples. Yeah. So this is the model. This is the loss value we trained during the training process. We can see this is the validation loss and this is the training loss. We can see the model is trained very well on the training set, but still get overfitted. So let's check when I just use them in practical. Yeah. So this is a training process still overfitting. Yeah. So let's see how it works. Yeah. So this is part of the data. If you'd like to see more data sets, you can just scan and you can see that. So this is the results just on the training data set. We can see the genome is just cut one time. Yeah, every chromosome is randomly cut one time or not cut. Yeah. So here, what we can see is that here, this is the genome after cut and shuffle. The results is here. We just cut here and inverse that. Yeah, this is the only modification we made, and we can see the model just find this error and modify this error. This is a very good sample to show that the model is working now. And how about this one? This one is a little bit more complex than that one. So we can see here there are some small fragments get cut and reordered and get shuffled. Yep. And then just after the model predicted the likelihood, we use the deep first search, and then we got this one. Yeah, works very well on the training set. And let's see another one. Yeah, this one we just every chromosome we just cut 3 times. So we can see in here, we got more small fragments. And after the model given the prediction, we ran the deep first search and we join them. We got this one, other one. Yeah, actually the answer is very good now, but still, this problem, yeah, has a little bit problem. We haven't just inversed them. If we inverse these fragments, this answer is perfectly right. Yeah. So yeah, this is all the results just on the training set because the training is very, very good. So it should give these kind of results. Okay. So let's move to the validation set. Yeah. What is really made us surprised is that actually the model also works on some samples from the test set. Yeah, the test set has never been put into the training. Yeah, and we can see here even after, yeah, this is the model after cut and shuffle. You can see here, these fragments should be moved. These fragments should be moved to here. All right. So we can see after the curation, the model works very well. Yeah, just here, only here. Here the model need to flip this small fragment, but the model didn't predict that well. And let's see more fragment, and this is the results from more fragments. We can see there are massive fragments. And after curation, yeah, at least although there are some problems here, but what I can see is that the model is very useful to cluster all the fragments which should be joined together. Yeah, but in some detail, it cannot solve all of the problem. But currently, the model is working now. So we can see all of the small fragments has been joined into a big chromosome. What our curators still need to do is to adjust the direction of the small fragments. Yeah. So let's see even more cuts. Yeah. For this one, the model also joined all of the small fragments to a big one. Yeah. So this is all of the results from on the test set. Yeah. Very amazing data set. Very amazing results actually. This really surprised us. Yeah. So and last, I want to say thanks to Shaoheng because this is a really a complex project. It's just from my project. Yeah, I'm getting salary because of this, but you're not. Yeah,
The
thank you
model.
so much.
Thank you.
Okay. So another thing I want to say thanks is Yumi. Yeah, Yumi gave me the high C data, and she works a lot for generating the high C map. And also I want to say thanks to all of the organizers for our hackathon and for the external AI. It is really amazing thing. Yeah. Yeah, for anyone is interested in the code we are using, you can just scan and download that and have a look. Yeah. Thank you so much.
Thanks so much, Sean. It's really good to hear that you've cracked through what I've seen you having a bit of trouble with for some time now. But so I was wondering, do you think that the problem was coming from the sort of landscape of the data? Is our genomes very what's the way to phrase this?
Yeah,
is it easy to overfit perhaps on genomes? Are they very, very similar in that sense?
Yeah, it is very easy to get overfitted. So just sorry about that. So just as we talked about the experiment, so if we just get the right order for the genome, but didn't give the enough information to it, or just give the noisy information to that, it can only get overfitted. Yeah, but actually this kind of data or this kind of cases is very, very frequent in the genome. Even in human genome, there are many such kind of, yeah. For example, if we just, for example, this is a human genome. So if we are going to just move all of the dense point to the diagonal line, this is not right, because this kind of signal, this one, is just a result from the repeat. Yeah. So if we just compress them and feed the data to the network, and the network may say, oh, this is the noisy at the input side, just like our experiment. So that's why the model is still overfitting now, and we are going to work further and try to solve or try to find a solution for that.
And do you think it's possible to extract the information of sort of how confident the model is, maybe before it's even made the prediction? Can you look at a genome and go, this is the right sort of genome for this?
Yeah.
Model, if that makes sense.
Yeah, this is very, very important, because sometimes when the joint likelihood of the model given is a lot very high, that means the model is not very confident to do that. So yeah, this is a very good idea, because yeah, this may help when we are going to find the order after we get the likelihood given. Yeah.
Any other questions? Oh.
I was wondering if the remaining noise could be related with your choice by only correcting the top, the highest value of the matrix. So if you return to your formal, like for example, slide 14 or 15, 14 maybe. Yeah, this one. So you would, when doing depth first search, you were correcting only the top one, right?
Sorry, what you mean top one?
The most likely.
Yeah, what you see is a very important problem, because in this deep first search, I just using the greedy algorithm for every fragment. I just select the best for him, instead of so for example, we have A linked to B. Yeah, I just find B is the best for A, so I linked B to A. But I didn't consider is that whether A is the best for B. I didn't consider that. This is also a problem for that, I think.
Also, I was wondering if there is a say. Also, yeah, so I was wondering if, for example, either doing like a wise first search, I don't know, I'm just, or you do multi-iteration of depth first search. So firstly, you connect B to A, and then to some reason by some means you merge A and B, and maybe try with C, something like that. It's very.
That's a very good idea. Actually, I'm considering to change the deep first search to broad first search. Maybe I can just compile all of the link first and try to link two of them maybe first and update the likelihood matrix and do that again. Yeah, but it is much more complex. We do not have much more time to do that.
Yeah, that's true. Thanks.
Thank you.
Any other questions?
Thank you for your presentation. It's quite interesting to see actually getting work done during this hackathon. I was wondering if it was adding a first actual layer just to see how likely it is for machine learning or for the model to automatically curate this data set. Because I'm thinking of if 20% of the data set are the hard one that requires potentially a manual curation, but 80% of them can be potentially cracked by this machine learning algorithm. Is that worth to first add a classifier to put these into the categories?
Okay. Yep. So thanks for your question. Actually, if the model is very well trained, yeah. If the model is very well trained, then for example for this one, here just have the small, very small problem. It will just give the likelihood like that. The likelihood matrix is very simple, yeah. So just to get this is small question here. So if there is no problem in this Hacking map, then the likelihood prediction will just be 0. There's nothing need to do. Yeah. So it is totally actually automatically to do that. Yeah.
I think it's more like on the entire data set level, because I guess the entire matrix is one experiment, isn't it? Or coming from one experiment. Just on that level, can you give machine to have a go on, oh, this is something the model can handle and this is not. And just to, I guess it might be difficult to train on the very hard experimental results with this machine learning model, because that might introduce the problems such as overfitting. But if the training sets or the evaluation in the end are relatively uniform, they are fitting more to your expectations, is that going to be a feature that a machine learning model could also pick up?
Yeah, it is a good idea to add this feature also into the model.
Mm-hmm.
Yep, thank you.
Great. So I think, well, thanks again, everybody. And I think at this point, we're going to have a discussion, but maybe it's best if we end the live stream and move out and have some drinks while we discuss.
Thank you.